Today the lecture is about proteins, structure and function of hemoglobin and myoglobin, types of the hemoglobin. In the previous lecture, we studied the catabolism of the heme and also the hyperbilirubinemia. Now the hemoproteins, structure and function of hemoglobin, myoglobin and types of the normal hemoglobin. Some important human and animal heme proteins. Actually, heme proteins that contain a heme prosthetic protein. They have a very large class of the metabolic proteins. They contain the heme group as a cofactors, which confers heme prosthetic group, which confers functionality of these proteins. Include the oxygen carrying, oxygen reduction, electron transfer, heme bound to the protein covalently or non-covalently. Some examples of the important heme proteins are the hemoglobin, myoglobin, cytochrome C, cytochrome P450, catalase, tryptophan, pyruvate. The hemoglobin and the hemoglobin has the transport function of the transport of the oxygen in the blood, while the myoglobin storage of the oxygen in the muscles. Cytochrome C, it involved in the electron transport chain. Cytochrome P450 monooxygenase system, hydroxylation of the xenobiotics, catalase, degradation of the hydrogen peroxide. Tryptophan, pyruvate, it involves in the oxidation of the tryptophan. Actually, the hemoglobin, you know it very well. Its function is the transportation of the oxygen from the lungs to the tissues and the transport of the carbon dioxide from the tissues to the lungs for exhalation. While myoglobin, the main function of the myoglobin is the storage of the oxygen in the muscle. Cytochrome P450, it is most important enzyme. Cytochrome P450 monooxygenase system, we call it. Most of the cytochrome P450 is to catalyze the oxidation of organic substances, metabolic intermediates like lipid, steroid hormones, and various steroid hormones and xenobiotics, toxic chemicals. So actually, cytochrome P450 is a monooxygenase reaction. For example, it involves in the insertion of one atom of oxygen into an organic substrate. While cytochrome C, it is associated with the inner membrane of the mitochondria. It is essential component of the electron transport chain, capable of oxidation and reduction. While catalase is an important enzyme, you study it very well in the topic of the enzyme biochemistry. It involves in the degradation of the hydrogen peroxide and convert it into the less toxic product. While the tryptophan pyruvate, also called as the tryptophan oxygenase, oxidation involves in the oxidation of the tryptophan. Now we discuss the structure and function of the myoglobin and hemoglobin. Myoglobin is the iron oxygen binding protein, primary oxygen carrying pigment of the muscle tissues. It also involves in the transport of the oxygen to the heart and the skeletal muscle. It is also used as a myocardial damage marker nowadays. It is reservoir for the oxygen. It is involved as an oxygen carrier, promotes the transport of the oxygen to rapidly respiring muscle cells. It has the high concentration of myoglobin and muscle cells allow organism to hold their breath for longer period of the times, just like the diabetic mammals. The human myoglobin consists of 153 amino acids. They are highly folded and compact structure with 8 separate and distinct alpha helical secondary structure, and its molecular weight is about 18 kilodalton. The heme is the prosthetic group of the myoglobin. Histidine involved in the oxygen binding with the myoglobin. Myoglobin can also bind with the carbon monoxide. Myoglobin is a single chain, and it is structurally similar to the single subunit of the hemoglobin. The interior of the myoglobin molecule is composed almost entirely of the non-polar amino acid. The rate of the oxygen diffusion from capillaries to the tissue is low because of the solubility of the oxygen. However, myoglobin increases the solubility of the oxygen. Myoglobin facilitates oxygen diffusion. Oxygen storage is also a function of the myoglobin, because myoglobin concentration are tenfold greater in whales and in seals than in the land animals. Next is the hemoglobin. Hemoglobin, it is a red blood pigment exclusively in the erythrocytes. Its concentration in male is about 13 to 16 gram per deciliter. In female is 12 to 15 gram per deciliter. In children, 11 to 16 gram per deciliter. Its main function, as we know very well, delivery of the oxygen from the lungs to the tissues, transport of the carbon dioxide and proton from tissues to the lungs for the excretion. Blood color red color of the blood is due to the hemoglobin contain iron, and when iron binds to the oxygen atom, it turns red. Hemoglobin structure consists of the this structure is alpha 2 and the beta 2 dimer containing the 4 polypeptide chain. The main function of the hemoglobin is the transportation of the oxygen. However, oxygen diffusion alone is too poor for transport in the large animals. However, there are the two alternative oxygen transporters, that is the hemocyanin, a copper-containing protein, while hemoerythrin, a non-heme-containing protein. Hemoglobin A, or the adult hemoglobin. Major hemoglobin in adults is composed of the 4 polypeptide chains, 2 alpha and 2 beta. They are held together by the non-covalent interactions. Hemoglobin is a conjugated protein. Globin is the hemoprotein part, while the heme is the prosthetic group. Heme is iron porphyrin complex compound with the tetrapyrrole structure. Here is the structure of the hemoglobin. Actually, students, the structure was already discussed in my previous lecture during the porphyrin chemistry and heme metabolism. Here the brief description again. The hemoglobin consists of the 4 tetrapyrrole rings. The outer carbon of the 4 pyrrole rings, which not linked with the methylene bridges, are numbered from 1 to 8. The methylene bridges are referred to as the alpha, beta, gamma and delta. Respectively, the 2 hydrogen atoms in the amino group of the pyrrole ring 2 and 4 are replaced by the ferrous iron, which occupy the center of the compound ring structure and establish the linkage with the all 4 nitrogen atom of the pyrrole rings. The iron, besides the linkage to the 4 nitrogen of the pyrrole ring, is also linked internally, you can say 5th linkage to the nitrogen of the imidazole in the ring of the histidine of the polypeptide chain. It is considered that the valence of the 6 as a ferrocyanide and the 6th valence is directed towards from the molecule is linked with the water in the deoxygenated hemoglobin. When hemoglobin is oxygenated, the water is displaced by the oxygen. The propionic acid group of the acid carboxyl group of the 6 and 7 position of the heme of the 3rd and the 4th pyrroles are also linked to the basic group of amino acid arginine and the lysine of the polypeptide chain. The central iron is oxidized and converted to the ferric state. It will carry the positive charge, which is balanced by taking the hydroxyl group from the medium. It may be balanced by the other anions like the chloride and the sulfate, etc. In addition to these hydrogen at the position 1 to 8, in pyrroles are substituted by the methyl, vinyl and propionic acid. In the structure about the globin, the protein part of the hemoglobin have showed that it is composed of the 4 polypeptide chain, 2 identical alpha chains and 2 identical beta chains. In normal adult hemoglobin, they are arranged in the tetrahedron manner. Each polypeptide chain of heme contains a heme and it is called as the heme pocket. Thus, one hemoglobin molecule contains the 4 heme units. In the normal hemoglobin, in place of the beta globin chain are replaced by other 2 delta chains, gamma chains or epsilon chain, and forming the different types of the normal adult hemoglobin. Now we discuss the types of the hemoglobin. These are the normal major types of the adult hemoglobins. Normal adult hemoglobins, these are the hemoglobin A, adult hemoglobin. It is composed of the 2 alpha chains and the 2 beta chains, and its concentration is about 90%. And hemoglobin F, fetal hemoglobin, composed of 2 alpha chains and 2 gamma chains, and it is almost less than 2% in the adult hemoglobin. Hemoglobin A2 or the minor hemoglobin or minor adult hemoglobin, it is composed of the 2 alpha chains and 2 delta chains, and its concentration is about 2 to 5% of the total hemoglobin present in the adults. While hemoglobin A1c, it is called as the glycosylated hemoglobin, and it is composed of 2 alpha chains and 2 beta chains, which is linked with the hexose moiety, and its concentration is 3 to 9%. Human hemoglobin A is just one member of functionally and structurally related family of the proteins. The hemoglobin, each of these oxygen-carrying protein is a tetramer composed of the 2 alpha globin polypeptide and 2 beta globin polypeptides. While hemoglobin F are normally synthesized during the fetal development, whereas others such as hemoglobin A2 are synthesized in the adult, although at lower level compared with the hemoglobin A or adult major hemoglobin. The hemoglobin A can also become modified by covalent addition of the hexose. For example, addition of the glucose forms the glycosylated hemoglobin derivative called as the hemoglobin A1c, and this hemoglobin is very important for the study of the diabetic patient. Fetal hemoglobin. Fetal hemoglobin or hemoglobin F, it is a tetramer consisting of the 2 alpha chains and the 2 gamma chains. The hemoglobin F is the mature hemoglobin found in the fetus and in the newborn, consisting of about 60% of the total hemoglobin in the erythrocytes during the last month of the fetal life. In the first month after conception, embryonic hemoglobin such as called the common hemoglobin 1 composed of the 2 alpha-like zeta chains and 2 beta-like epsilon chains. You can say 2 epsilon chains and 2 zeta chains are synthesized by embryonic yolk sac, and governed hemoglobin 2 has 2 alpha and 2 epsilon chains. Within the few weeks, liver begins to synthesize the fetal hemoglobin in developing bone marrow. Hemoglobin adult, that is hemoglobin A, synthesis starts in the bone marrow at about the 8th month of the pregnancy, and it gradually replaces the hemoglobin F. However, the hemoglobin F represents less than 2% of the total hemoglobin of the adults. The most important feature of the hemoglobin F is its binding with the 2,3-bisphosphoglycerate. Under the physiologic conditions, hemoglobin F has higher affinity for the oxygen than does the hemoglobin A or the adult hemoglobin. The gamma globin chains of the hemoglobin F lack some positively charged amino acids that are responsible for binding to 3-bisphosphoglycerate in beta globin chains. Weaker interaction between the 2,3-bisphosphoglycerate and hemoglobin F results in a higher oxygen affinity for hemoglobin F as compared to the hemoglobin A or adult hemoglobin. The higher oxygen affinity of hemoglobin F facilitates the transfer of the oxygen from the maternal circulation across the placenta to the RBCs of the fetus. Next is the hemoglobin A2, a minor hemoglobin. Hemoglobin A2, a minor component of the normal adult hemoglobin, first appearing shortly before birth and ultimately constituting about 2% of the total hemoglobin. About 2 to 5% of the total percentage of the hemoglobin is the hemoglobin A2. It is composed of the 2 alpha chains and the 2 delta chains. Next is the hemoglobin A1c or glycosylated glycated hemoglobin. Hemoglobin A is slowly and non-enzymatically glycosylated. The extent of the glycosylation depending on the plasma concentration of the particular exos. Hemoglobin A1c, it has a glucose residue attached predominantly to the amino group of the N-terminal valine of the beta globin chains. Increased amount of hemoglobin A1c are found in RBCs of the patient with the diabetes mellitus. Next, here is the globin chain with the synthesis of the globin chain and the hemoglobins. Alpha gene family. Here is the alpha gene family. There are 2 genes coding for the alpha globin chain present on each one of the chromosome 16. The zeta gene and the other member of alpha gene cluster is also found on the chromosome 16, is active during the embryonic development. While the beta gene family, the synthesis of the beta globin occurs from the single gene located on the chromosome 11. This chromosome also contains 4 other genes. One is the epsilon, expressed in the early stages of the embryonic development. 2 gamma genes synthesize gamma globin chains of the fetal hemoglobin. One is the delta gene producing delta globin chain found in the adults to minor extent, that is the hemoglobin A2, and beta gene two beta genes which are involved in the formation of the adult hemoglobin, which is the major hemoglobin in the adults. So zeta and epsilon 2 involved in the proper hemoglobin formation. Gamma 2 and the alpha 2 involves in the formation of the fetal hemoglobin, and alpha 2 and delta 2 involved in the minor hemoglobin formation. Hemoglobin A2 and while alpha 2 and the beta 2 are involved in the major adult hemoglobin, that is the hemoglobin. Here is again the representation of the synthesis of the gene globin hemoglobin, zeta 2 and the alpha 2. Sorry, zeta 2 and epsilon 2, gamma and alpha involves in the synthesis of the fetal hemoglobin. Fetal hemoglobin consists of the alpha 2 and the gamma 2, while the adult hemoglobin A2 is synthesized by the alpha 2 and the delta 2, and the adult hemoglobin, that is hemoglobin A, are synthesized by the alpha 2 and the beta 2 genes. And here is the molecular representation of the synthesis of these globin genes. Here is the globin, alpha globin and the beta globin gene families containing the 3 exon coding regions. They are separated by the two non-coding introns. In the transcription, there is a formation of the premature messenger RNA, which become mature after the splicing and the removal of these exons. And this mature messenger RNA then from the nucleus enter into the cytosol, which involve in the transcription reactions and involved in the formation of the protein, that is the globin protein. This was all about the structure, function of hemoglobin, myoglobin, and the normal types of the hemoglobin, also some examples of the heme protein and their